I would like to briefly talk about the Data Coordination Center. And then I think we already said that. Then please post your questions in the Skype chat for the streaming. So please post your questions in the Skype chat. That's fine, and then we will try to answer. I think you have not asked questions so far, but please do it. It's actually good if you ask questions. I encourage you. So this is just I think I'm repeating what Ferenc has said, but this is the big frame of the DANIO-CODE Aims, and the first one is the establishment of the Data Coordination Center. So this is what I will talk about now, the first point here. And some of the key points from the DANIO-CODE document are listed here. It's a standardized data input, shared data resource, centralized access, and so on and so on. So I will not read all of this. Instead, I will go on and say what are maybe typical questions that we would like to enable us and the DANIO-CODE members and also the rest of the world to ask. And for example, questions on the data availability. So because we're collecting a lot of data, so the first thing is that you can ask, for example, you would like to know if there is experiments of a specific type, whatever, like a specific knockdown experiments available for a certain gene. You can ask that. And if so, you can ask what kind of data is there for those knockdown experiments. There's chip data or RNA-seq data or similar things. You can also ask what kind of chip-seq data sets are available for a certain epigenomic mark or for a certain transcription factor. You can also ask other things like what is in general available for a certain fish strain. Show me all the data that's available for a certain strain. You can also ask if you are interested in comparing two types of data and you want to, say, develop a method for doing that. You could ask for which data sets are, for which samples are those two types of data available. Or the last one would be that you would like to know for the experimental background of a certain experiment. So you find a certain experiment and you would like to know the detail of how this has been generated. You can also ask this question. Another type of question is not about the data itself, but it's about the genome annotation, which was done based on these data. And so for example, for me a typical question would be how many promoters are there for a certain gene in the fish. Often it's not one, but several. And then you would like to know where's this gene expressed specifically, and maybe where's that specific, is there a promoter specific expression for a certain gene in a certain tissue or in a certain genomic background or a certain developmental stage. You might want to ask like some half analysis questions, like half the way. You would like to know if a certain gene is differentially expressed between two different tissues. Or if the genome, what is the genome conformation around a certain promoter of a certain gene? Or which are the transcription factor binding sets that are present in the promoter of a certain gene? So this is already kind of derived information. Then our overall concept for the DCC, for the Data Coordination Center, is depicted here. So there's, say, 3 big blocks. The block in the middle is actually where the data is living. So this is the database and also a file repository. I will talk about some of these also in the next slides. But so there's the place where the data is living, and then there's another part which is the green one, which is the input. So what we are entering into the database are 2 things. One is the data itself, and then also we need to enter the description about how this data came about and so the annotation about the data. And all of this is going into the database. And then we would like to look at our data in different ways, and this is what we call a view. So you can then look at the database in different ways. You know, you can extract data with a Biomart type of framework, or you can use a genome browser, or you can use any kind of creative visualization that you can imagine. I will maybe go into some of these in a few slides. So the overall workflow is then that all the collaborators of DANIO-CODE are sending data to the DCC, and then we, the DANIO-CODE members, they are also, I call them users here, but the DANIO-CODE members, they are annotating or they're describing the data. So this is what we are doing today. And then there's a lot of things that we have to do, which is that we have to perform the standardized data processing. We have to introduce the data into the database, actually. And we have to perform the genome annotation analysis. So we are not annotating the data, but we are performing the, some information that is derived from it. So we are identifying the genomic elements from these data and so on and so forth. And another thing is that we are working, as Ferenc mentioned, working together with ZFIN, and we want to use their tracker. So we will synchronize all the data that we generate and all the derived annotation with the ZFIN or ZFIN. So the ZFIN will be one of the accessibility points, so where you can actually get these data. It's not the only one, but it's one of them. And then actually, from the perspective of a user, then after this has been performed, these steps, then you can actually look at your data again and also at the data annotation. And that what this probably means is that there's actually quite some waiting time. So this is a bit of a, not a warning, but already, so I guess it's good to understand that after we have annotated the data today, actually there's a lot of things are happening so that before you can look at your data for the first time and at the genome annotation which is derived from it. So I'll briefly go over the individual points. So the data transfer, I think you have seen that we asked you to ship hard drives or USB pen drives or so. And actually there's very few, we did testing and we found out that it often didn't really work to transfer the big data sets to, like, over the internet. Yeah. So, but we are, I don't know, I think we will keep testing, and for some locations it actually worked. So the data annotation logic, I would like to briefly explain what we had in mind with this one. So how do we describe the data itself? And Matthias will show this in detail later on. But the overall idea is that we said, everybody has data sets in their hands, right? So you have a FASTQ file or a Wiggle file or whatever, and that's the unit that you are looking at. And so it feels if you just say, in the first case, it feels very much natural to look at these things and then to say, okay, I just look at these files and then I describe them each by each. But actually, what is, of course, like many of these experiments have been done with a global question in mind. So we said that we rather do it the other way around, so we take a top-down approach, where we first describe the experiment as such, the big experiment. So which we call the series, which is on the top right here. So we describe what did we have in mind when we designed this experiment. It's the overall contains the overall experimental design. So it's from top down, and it's from general to specific. So as you go from top down here, you become more and more specific. And this is important to understand the experiments, the individual experiments that have been done, and to put them, be able to interpret what you see later on. And this also helps to, it also is very important then to use a very standardized vocabulary, so you can compare this experiment to another experiment. So for the database, we have different types of databases, which you usually don't see so much yourself. One of them is a file or a data repository where all the FASTQ files go in. And maybe later on also some BAM files, BED files, or Wiggle files. This may be a flat file structure. And then there's an annotation database which contains all the annotation of that data. And this is just a screenshot. You cannot see it in detail, but this is the database structure of the data annotation database. And then the data will also be available in ZFIN track hubs. To be able to make the data comparable, we want to process data in a very standardized way, and or in a very uniform way, and Ferenc already mentioned that. So for example, all the chip-seq data you have in the whole data repository, you want to have the p-calling made in the same way, you want to have the mapping before made in the same way, the RNA-seq, so the RNA-seq data or the cage data or any kind of epigenetic data you have. And we started a discussion group for how this data processing should be done in a standardized way. Because when you look at it, then you see that there's many different ways of how you can process the same data, and people have different opinions. And since this is a community effort, we would really like to get the input from all the DANIO-CODE members. So we would like to hear what you think about the data processing, and if we can, or how we can agree on the denominator, how we can agree on the data processing. So the pipeline discussion group has been initiated, and I think one of the students, Kadir, has sent emails around and asked the DANIO-CODE members to join if they want. And this offer is open all the time, so we're happy if you join and contribute to that pipeline discussion. Then just a comment on the views. So I think the most obvious views are a view on the data, and I just kind of was borrowing a screenshot from the ENCODE project here. And you cannot really read it, but it says in the rows you have like different cell types, and in the columns you have different data types. And then you can see basically as a table of an overview what kind of data types are available, and then you can obtain them if you want. Just as a table. Of course, then there is also other views where you can ask really clever questions, and you can basically refine questions and get data sets based on your questions. That's called the BioMart system, or a BioMart system is a way that allows you to ask clever questions and to download data based on the criteria that you are setting. Another way to visualize is then also a genome browser view. That just took a random genome browser here. So you see then how not the original data you uploaded, but how the genomic elements that are identified from the data, how they are visualized in the genome browser. And one important thing to mention is that we that we also want to develop creative new views. You could, for example, imagine, or one of the strengths of the zebrafish data is that we have a lot of the developmental time courses. And so you can imagine that you have like a lot of data types on these developmental time points. And I don't have a picture for that, but you can imagine that you want to find new ways actually of showing this. I forgot to include a slide here actually that I wanted to show. But so we want to work on creative new visualizations of how to put the type of data we have for the zebrafish. So that's one of the strengths of the types of data we have for the zebrafish. How to use that in the best way. This is my personal guess of a timeline. I don't know, so don't hold me completely responsible for it later on. But the the idea would be that we have our first data annotation jamboree, which is in May now. And then basically we want to start with the data processing hopefully soon. So over summer we want to basically process data, but depending on the pipeline discussions, maybe we will start with the data types where we have an agreement. Some of the data types might need more discussion. And so we can already start with the first data views in tables or in genome browsers during the summer. After we have performed the data processing, we would like to give a feedback to the data producers, which is the DANIO-CODE members who have actually uploaded the data and who are annotating it today. And we would like to, I think, perform something like a sanity check, I guess. So we would like to ask you to look at your data and see if this is what you know, if we did something wrong, or if you recognize your data, if it looks good to you. And after the feedback, we would then go ahead and perform the genome annotation, and then we would visualize the data for the whole consortium. towards autumn. And then we would also start, actually, at least one of the PhD students will start to create novel views on the data and so on and so forth. So we hope that by late this year we would have the first data release. That would be one of our goals. Then whenever you have a continuous effort of collecting data, then you would like to have checkpoints in that. So we call these checkpoints data freezes. And so a data freeze would be something that we collect the data, and at a certain point of time we say that we process these data now and we perform the genome annotation based on these data. So that's the first data freeze. And most likely I think we would basically start freezing the data after the Jamboree. I think if you're like a day or two late with your annotations, maybe that's also fine. But basically, like a few days after the Jamboree, we would like to freeze the data and the corresponding data annotation so we can go ahead and start the data processing. But this will, since the data collection, we want to encourage you, of course, not to give up afterwards, but to continue and actually have additional labs to provide the data. So this will be a continuous effort. So we will continue to include data, and I don't know, depending on if we have a like significant amount of additional data collected, we will perform another data freeze, and then we will perform another annotation round. And I think we have no understanding yet about these update cycles, so often they would happen, but I think we have to learn as we go. Ferenc has already showed the overview, and you cannot see it in detail, but what you can do is you can go on the Wiki page, actually. That's very important. So you can see what kind of data we have, and that may actually also encourage people to put additional data, because if you see how you know how others are uploading, then maybe you also would like to upload more. So for now, I wanted to show the two references here, and I think maybe we are sharing the slides later on the Wiki. But anyway, so the DANIO-CODE Wiki is a very good place to look at, and also there is the guide document for the data for the DCC, so you can see how you can read in detail how data annotation will be performed. And very importantly, I would like to acknowledge the people who are involved. So the people who really have done a lot of the work are like in implementing technically is Matthias, Kadir, and Marcus. And then advice from Ben and from Boris and from Dunja for testing, and also from Ferenc for giving us the vision and really for holding us together in the consortium. That's very much appreciated, Ferenc. Thanks a lot. All right, I'm done.